So this lab is going to discuss a little bit about, the video will discuss a little bit about the lab, which is qualitative analysis of anions. And there really isn't much to talk about. That's a big difference from what you've done for the other two qualitative analysis labs for cations. The only difference here is that with the anions, most of the reactions are fairly specific for a particular anion. And what that really means is that you are able to carry out the reaction in the presence of all the other anions. So in other words, you don't really need to separate them first like you did for the cations. Now this is going to be true in most cases, but it's not going to be true in all cases. So in some of them you do need to perform some kind of separation to get rid of some of the other anions if you think they might be present before you go ahead and do your specific confirmation test. But again, in the case of the anions that we are going to work with in this lab, most of them have reactions that are specific enough to just identify one anion in the mixture of many anions. The other thing that's a little bit different about this particular lab, as far as control is concerned, is that in your positive control experiment, before you always just make one set of positive control and you carry out the test to make sure that you know how to do the experiment correctly. In this case, there's going to be two positive controls that you're going to make, and they're really the same solution. It's just that one is concentrated and the other one is diluted. Okay, so the concentrated control is just basically the solution that you will take out from the stock bottle, from the reagent bottle in the hood. And the diluted sample is what we refer to as a tenfold dilution, which really just means that you're going to take 1 milliliter of the stock plus 9 milliliter of water. Okay, and that's going to give you your diluted sample. Now, when you actually test them, you're going to use only 1 milliliter of stock plus whatever you need to run your tests. And for a diluted sample, same thing. Even though you might make 10 milliliters here, you're only going to take 1 milliliter to do your test. So it'll be 1 milliliter plus whatever you need to do to add to it to look at your test. The reason why we do this the following way, to have two different concentration of the control, is because the unknown itself actually has a mixture of anions. And in the unknown, the concentration of the anion is usually somewhere in between these two values. Okay, so as a result, you're doing qualitative analysis, so you don't know exactly how much of the anion are present in the unknown, but if it's somewhere in between, then the observation you're going to make will depend on knowing what the extreme values are, where the concentrated value and what is the diluted values, right? So if you've seen those two observations, then you should be able to know what you should expect to see in your unknown. Okay, so that's the whole point of making both of them, because the problem is if you just look at the concentrated and you get something that's a lot less than that, you might think that the anion is not present, but it's actually present because this value looks similar to the dilute sample, for example. Okay, so that's kind of the whole idea of making these two controls, so you can have ranges, of observations between this and this, so that you know that the unknown should be somewhere in between, or maybe closer to diluted, depending on how many ions you have in your solution. Okay. Again, lastly, I just want to repeat the idea that most of the anions could be tested directly without separating them from each other. However, some of the anions do require separation, and these are indicated in your lab procedure. So when you read them, just make sure that you look where it says, your potential problem might be you can have other anions that might interfere with the test. That would generally mean that you would have to separate the interfering anions first before you can actually carry out your test.